there are notes from last week still there. Recorded session. If you're interested, we'll do the same this week. Start recording in a few seconds so that you can share with your friends. Here's the material for this week. I'm going to just change this a little bit. I want to have more of my browser available to me. Yeah, and so now we're going to do this week. We'll tackle round two of our workshop, and I'm interested in. I'm not sure that I've scheduled things exactly right, so you have to tolerate me a little bit. But I kind of wanted to accomplish two things. One was kind of explore more of the AnVIL and what's going on in that environment, and then I also really wanted to highlight the AnVIL package, which is an R package that makes working with AnVIL, the overall ecosystem, pretty convenient. So we'll see how it all works today. That's basically my learning objectives: learn a little bit more about what goes on in AnVIL, and then learn about the AnVIL package. You'll see, of course, that we'll use AnVIL. There's this demo script in R that you can open up, and we'll follow along in the second half. We'll execute those commands. It'll be convenient to be able to follow those. We'll look at a couple of workspaces that are sort of mature, and in many ways it'd be totally awesome to delve right into these workspaces. Actually, Kayla, I think we should probably start recording if we haven't already.
Yep, we're all set.
Oh, we're good. Great. Excellent. So we'll delve into these workspaces, and then the AnVIL package. If you're not really familiar with Bioconductor packages, they've usually got some great documentation in the form of vignettes that are available from the website and from the package. And this is a AnVIL package vignette, which goes into quite a bit of detail about different functionality that we'll talk about today. So it can be a really useful resource for you. Just to review what we've covered last week, we basically did the essential steps of logging in, navigating to a workspace, billing accounts as a way to pay for your computing, and then we introduced cloud environments, both the Jupyter Notebooks cloud environment and the RStudio cloud environment. And with those cloud environments, basically you can do anything on the cloud that you can do locally. And we'll spend today using RStudio just because it's more familiar to me and I relate to it more, even though maybe in the AnVIL itself, Jupyter Notebooks are more mature and supported. RStudio still has some areas which aren't quite so integrated, but we're going to focus on RStudio. And then when we talked about these cloud computing environments, we learned about the runtime, which is like the compute power that's doing the computation, and these so-called persistent disks. And then maybe just to highlight the cloud computing environment, like our RStudio instance, is something that's individual to us, and it's not really shared with others in our lab or whatnot. We use the workspace as a way of sharing our work. And to a certain extent, the cloud computing environment is ephemeral in the sense we can destroy it and lose the material that was perhaps saved there on our persistent disk or whatnot. So maybe we can think about wanting to have a more persistent environment. So a couple of little comments there. There are some great questions asked at the end of the last session, and I've filled out the fact of the notes to answer those questions. I did a little bit of research. One is that the persistent disk is mounted at this location on the Jupyter Notebooks and at this location on the RStudio image, so just slightly different. And when we saved empty cars, that file to this location, and then went over and expected to find it over here, we'd actually lost it. So the persistent disk is mounted in a slightly different location. And we also talked about customizing our Docker file in, sort of sudo-like access. And there are actually two ways of doing that, either writing your own Docker file or writing a little startup script. So the facts have been answered. So that's kind of the review. Maybe take 2 seconds if people have any questions they want to chime in with. All right.
I had one question. Martin, when you said the file was lost there, do you just mean you have to look somewhere else for it, or you actually can't find it when you created it in Jupyter and then looked for it in RStudio, like it's really not available?
It's really not available. It's like it's stored with the runtime and not with the persistent disk. And we switched from Jupyter to RStudio, so we switched runtime, so it was gone. Yeah. So really, you'd like to store things in the Jupyter notebooks. Under the notebooks tab is where the persistent disk is located.
Thanks.
Yeah, it's a little bit surprising, and I mean, we did have a little bit of discussion at one of our technical calls last week about that, and maybe ways of making that a little bit more robust or improving it in the future. So at some point it'll probably be better. Okay, so if you've registered for the workshop, we should be able to get going. You should be able to, we're going to log into AnVIL, and we're going to go and find the Bioconductor workshop pop-up. And then if we had one from last week, we're actually going to delete it. If we had a version of the pop-up workshop from last week, we're going to delete it and start again with a new clone of the Bioconductor workshop pop-up. So we'll just make a new clone, name it uniquely for ourselves, and then we're going to use a slightly different flavor of billing account, the May3 billing account. And then we're going to start the cloud environment. So let's just do that. And I hope you'll follow along with me on your own computer. We'll go to AnVIL, anvil.terra.bio, and then go to the hamburger and choose to sign in with Google and sign in with the email address that we used to register for the course, for the class. And then go to Workspaces, and you should see this Bioconductor workshop pop-up. It should be accessible to you. And if it's not, just jot a note to Kayla in the chat with the address that you're trying to use, the account that you use to log into AnVIL, and she'll make it available to you. If you had your workshop from last week, you made a clone last week, you can go here and choose delete. But what we want to do is Bioconductor workshop pop-up clone, call it something unique. So I'm going to call it MT Morgan. And then select a billing project. And this week you want to choose the billing project May 3, Deep Pilot Bioconductor May 3. And clone the workspace. So hopefully this is like kind of reiterating what we did last week and kind of familiar to you.
Do we want to do anything with the authorized domain select groups?
No.
Or? Okay.
You don't have to. Yeah. It's kind of fun.
Thanks.
Yeah, it's good. All right. Yeah.
Martin,
So
what was the billing project again that we should choose?
You should have access to a billing project called Deep Pilot Bioconductor May 3.
Deep Pilots, yeah. Thank you.
May 3. Yeah. Actually, I should say that last week we spent all of $1.08 on our computing, so it wasn't expensive. We'll try and build up the bill. Each week we'll try and do better on spending money on our grant. So then we wanted to, hopefully we've got to this workspace. I can't see the chat, but if you're having trouble, maybe you could just make a note there and Kayla will let me know or speak up. Awesome. So now we wanted to associate an RStudio instance with this cloud computing environment. So I'm just going to open up, click on this cloud environment, which currently says none. And then I'm going to go to this customize location and choose customize. And in this drop down, I'm going to scroll down until I find Rstudio. So, and then again.
Sorry, I just missed you. How did you get to this one spot here?
Yeah.
Oh, okay. I'm at dashboard. Okay, I'm in the same spot you are now. Sorry. Oh, there it is. Okay, now I see it. It was hidden before with the chat box. Thanks.
I see. Cloud environment. And then this customize. And then Rstudio. And then, as before, we could adjust our compute needs, but we're going to stick with exactly what the defaults are. A single CPU, not very much memory, and a fairly small persistent disk of 50 gigabytes. And we'll just click create, and Google will run off, and up here it says cloud environment creating. So it'll change from creating to running or something like that when it's ready. But right now, Google is going out and finding the resources to satisfy that request for me. And while that's going on, it's like one of those cooking shows, you put the turkey in the oven and then you go away to a commercial break and come back and the turkey is ready or whatever. So I'm going to go back to these notes here. And we started our Rstudio cloud environment, blah, blah, all those steps. That's great. Launched it. And we're waiting for it to be running. But we won't quite get there. So what I wanted to do was, while we're waiting, I'll go back to my workspace, and I actually wanted to explore one of the other workspaces that we have available to us. So I'm going to click on where it says Workspaces here. I'm going to click on that. It's a kind of a shortcut. I'm going to click on the big Workspaces in dark color. And you'll see that there are several different types of workspaces. I'm going to switch over to the public workspaces. And then in this search workspaces, I'm going to look for HCA, the Human Cell Atlas. So the Human Cell Atlas is a project to generate single cell sequencing data for individual cells from different parts of the human body. And there are a large number of data sets that are becoming available for them. And I'm going to look at this workspace called HCA Optimus Pipeline. And I encourage you just to follow along here. We're just going to look at it. We're not going to do any computation. You don't have to clone it. You should have reader access to it, so you can just look at it and see what they're doing. And so this is a workspace that was developed by some folks at the Broad Institute, and it takes some sequencing data, single cell sequencing data like the FASTQ files, and then it transforms the FASTQ files through a series of steps, a standardized pipeline, into a file that contains the count matrix of individual cells, those 60,000 cells, and individual genes in those cells, and the measure of expression of each gene in each cell for the experiments that we're involved with. That's what the workspace does in broad ways. It's kind of fun, right? Like we've got this data and it's going to be processed in a standard way. We'll see how we do the data analysis here. I'm going to pay no attention to these error messages that keep showing up. We're not actually going to do the analysis, but we're kind of orient ourselves. We'll do an actual workflow next week. So here's our workspace, and I'm going to look on this data tab. And we didn't actually look at the data tab last week. We looked at the notebook. So data, it says that the person who created this workspace, which you and I might do as we're becoming more proficient, created a series of tables that summarized the samples and the participants that are going to go into this project. So if we click on samples, you can see that there are 10 different samples. They all have an ID associated with them. And then each of the samples has these FASTQ files, which are like the data that was generated by the sequencing technology, the Optimus pipeline. So, and if you mouse over one of these guys, you can see that it pops up and it says gs:// blah, 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 ending in fastq.gz. So we know that it's the FASTQ file that's come up. And then the GS means that it's the person who made this workspace has put the data into a bucket. It's called a bucket on the Google Cloud. So this is like a place to store information on the Google Cloud. And the bucket could have permissions. It could be visible to everyone, or it could be visible to only you or to your lab group or whatever. So the FASTQ files have been put onto these Google buckets, and this table summarizes the samples that are involved in this, that are available for this workspace. So we've kind of coordinated a little bit about the samples that we're going to use, and then the FASTQ files that are involved. And then this sample set table is a little bit tricky, but actually what it does is that if you scroll over here just a little bit, what it does is it groups those samples into sets. So this, we had 10 samples over here. So 6 of the samples are put into this one set, and they all turn out to be a particular, the neurons 2K. They're all samples from one part of the experiment. So the person who created this notebook has taken these samples and divided them into individual sets corresponding to sort of coherent groups that are supposed to be analyzed. So we've got these sets. And now what I wanted to do was I wanted to switch over. So, okay, this data tab contains these tables that somehow have information about the experiment that we're going to analyze. And it kind of looks like a kind of familiar CSV file, and we can kind of navigate over this in in different ways, visualize it, and so on. So that's kind of available there. You can see that there are these graphical user elements, but actually they're not very useful to us. We'll find that there's a much better way of dealing with these things. So we've got these data tables. And what I wanted to do was plug the data into a computational workflow that transforms these FASTQ files into count matrices. And you can imagine that this is actually a computationally intensive operation, that you have to do significant amount of computing to go from a FASTQ file to the count matrix. So I'm going to show you the overall structure of this process, but we're actually not going to do it. So it's a little bit abstract, but next week we'll do a workflow a little bit smaller and manageable. So you can see under this workflows tab that we've got a bunch of, the person who put this together has provided a bunch of workflows, and these come from a workflow store called Docstore. We could find a workflow or add our own custom workflow, but I'm gonna just go with one of the workflows that they've provided us with, this Optimus Human V3. And click on that. Hope you're following along. And so the workflow consists of scripts, inputs, and outputs, and then this magic button that runs the analysis. So when I look at the script field, I get the substance of the workflow that's consists of a series of tasks that maybe you can imagine what they're supposed to do in the workflow. Like this one is star align BAM. So it's going to take the FASTQ files and align the individual short reads to a reference genome. And you can see tag sort BAM and UMI correction. So these are like steps in the workflow that will perform UMI correction, a step in the analysis of the FASTQ files as you take them through alignment and tallying of the number of individual reads in those FASTQ files that align to particular genes. So we've got like a bunch of steps, and then we've got like this description of what the workflow is, like a definition. It says, this is what the workflow does. It takes a bunch of inputs that include the FASTQ files and the reference genome, and then it performs a series of steps. It calls the input check, then the FASTQ processing step, and then the modify GTF step. And then this one is kind of interesting. That calls the star align BAM, and it says scatter, which means for each pair of FASTQ files, separately perform the alignment. And all of the computational resources are going to be managed for us. We'll just end up pressing this magic button. But what will happen is that each of the pair of FASTQ files will be separately aligned, and then the next steps of the workflow will carry on all of these steps that someone has put together.
So is scatter applying like a multi-threaded process per node, or is it deploying it across multiple nodes when it kicks off scatter aligner?
Yeah, it's actually going to launch, like each pair of FASTQ files will get its own compute node in the Google Cloud. Yeah.
And how is it designating how many nodes to give this whole data set?
Well, so we'll see a little bit when we write the script, we can say what the compute task for this particular task, this star align single end. If we delved into it, we'd see that it said that the person who wrote the script thinks that this should require X gigabytes of memory and Y CPUs per node. So it'll allocate resources that way.
Thanks.
We'll see a little bit of a simpler example next week, but those are great questions. And also, for you and I as end users, it's completely transparent. We don't know anything about the compute resources that are being required or anything like that. The person who wrote the workflow has figured that all out for us. And then at the almost the last step, I think, is this Optimus Loom generation, which generates the output file that we're actually going to be interested in for our interactive exploration. So we go through a bunch of steps. All of the compute stuff is taken care of for us. And then we get some outputs, including the Loom output file that we're going to be interested in.
Are there any options to change the compute resources at runtime? Because I would imagine that depending on like the size of your samples, that it might change how many compute resources you would need. If you had like really low coverage data, you would need a smaller instance than if you had really high coverage data.
Yeah. So there are a couple of things. It depends on how if the person who wrote this script had that in mind, they might have parameterized it. Like one of these inputs might be, how big a compute node should I use? But if they haven't, the script is actually it's in GitHub, and you can modify the script to your heart's content, and then run it yourself. You're not restricted to using this. This is like a script to get going, as opposed to the be-all and end-all. So the script has inputs and outputs, and the inputs tab kind of wires the data that we're working with to the script. So you can see, for instance, that the script requires the FASTQ files, and it says the FASTQ files are to come from the samples table and this column in the samples table. And so that's like the R1 samples, and the R1 FASTQ files, and the R2, the second end files, come from the samples table and this column in the samples table. And then likewise with the outputs, it says this Loom file is going to be added. You can see up here it says tables sample set. The Loom file is going to be added as a column in this, which refers to the sample set tables, as a column called Loom output file. So we've kind of we've got the script and we've wired the inputs from the data table and the outputs also in the data table. And then we're going to run the analysis, and a time will pass. This would be a computationally expensive thing, but the person who created this notebook has already run the analysis. I should say, here you can see what happens when you run the analysis. You get a job that records who's running the analysis, and this is its eventual fate, but it goes through stages like, queued and submitted and running and broken. And then you can see here, I clicked on this green, the top one here. You can see that the job last ran in January of 2021. It succeeded. And then this little dashboard, workflow dashboard, shows us each of the steps. And actually, if we were actually running it, each of the steps would be updated dynamically, telling us the step had succeeded and so on, eventually getting to the final step, succeeding. And of course, this is like I don't know, optimistic, because usually when you run your workflow, something goes wrong, right? And so you'd actually drill down here. There would be a green check. There would be a red X, and the red X would say, when you got to here, it would say, oh, I got through 7 steps, and then something terrible went wrong at this stage here. And then it's like figuring out, there'd be links to the output, and you could determine what was going wrong with your script. Yeah.
Question, Martin. If we wanted to modify the script, would that look like just a fork on GitHub, or would we have to make a new project on Docstore?
Yes. So probably what you do. So we could drill down a bit on this. I'm not exactly sure. So I'm gonna look at this. I'm not sure where it's gonna go. So if I get too into the weeds, we'll stop. But so I'll just go to Docstore, and Docstore is like this collection of scripts, and straightforward to create an account. And you can see it says this is maybe it's a little bit small. It says the source code is actually from this GitHub repository, and if you open that, you could see this is the GitHub repository, and I think we're talking about tasks, and so each of the tasks are this is where the script is actually implemented, the task is actually implemented, and so you could imagine that you forked this repository, made changes to the script to customize it, but then you'd probably also have in Docstore you would have registered yourself and would have pointed to your repository as opposed to the Broad's repository. And then when you ran the script here, you would have find a workflow, you would have navigated to your repository and done that. So I guess that's the guts of it. Complicated, right? So we should focus on the amazing power that we could actually run one of these workflows ourselves right now and actually have it transform those FASTQ files into a Loom file. We should focus on that kind of level of amazingness. And we could probably have pretty good success if we had our own data. We could probably alter this data table to point to our data, and probably the pipeline would work. It's probably been debugged and robust enough that it would work as a first pass anyway.
Martin, just to kind of follow on for Levi's question there. So most of the data we get is multiplexed, and so I didn't think this was set up for multiplexing or for site-seek. So if I wanted to edit this workflow to add in those tags, I would have to do it via Docstore or GitHub, or would I just do it actually on this space?
Well, the first thing is that there might be, I don't really know, but there might be a better workspace to start from. So like, I was thinking about, well, if you're doing, say, a tax seek or something like that, that perhaps there'd be another, workflow that's already been constructed. You're kind of asking a kind of,
That's useful to know.
Yeah. Yeah, so like this was just somehow an arbitrary workflow that was complicated, but I hope relatable enough. But you might end up, I mean, if you're writing your own workflow, you're probably becoming familiar with the workflow description language. We'll see an example next week that's not nearly as intimidating, where you'll write a workflow description language, probably in your own GitHub repository, probably referenced from Docstore.
Thank you, Martin.
Yeah.
I just noticed this cool button there, Launch With. So I guess once you put your project onto Docstore, you can use that button to load it into an AnVIL workspace.
Yes, this will either create or ask you to add to an existing workspace. So that's right. Yeah.
Can I ask a stupid question there? There's two different buttons. One is Terra and one is AnVIL, and I've been using the two kind of side by side with each other. If you did one button versus the other button there, do they go to different places altogether, or it's?
Not different places altogether. One of the facts that I synthesized as a fact from in last week's was about Terra versus AnVIL. Terra is like a generic version of AnVIL, and AnVIL is like a branded skin for the NHGRI. So they actually have very similar functionality, but for instance, your account was an AnVIL account and not a Terra account. So you'll click the AnVIL button.
Martin, while you're answering questions, can I ask one maybe a bit simpler? So when you're back in your workspace or back in your thing, so you launched a cloud environment as I did, with the Rstudio and R running, but up there it says cloud environment none.
Yeah.
But in my other tab, because I opened a new tab to do it, I see the R environment running. So if you did accidentally load it, how do you get back to it, or what's the best way to navigate between environments you might have running? And you can run multiple environments simultaneously, et cetera, et cetera.
Yeah. So actually, that's great, and it's a little bit evolving. So it used to be that, like say a month ago, that the runtime was sort of common across all of these workspaces. So the cloud environment over in this featured workspace would be the same as in your pop-up workspace, but the runtimes are being aligned with workspaces. So that's like the vision. And actually, I wanted to go back. That's perfect. I wanted to go back and go to the my pop-up workspace, and here's my runtime, which is now available. And I'll just open it. I hope you guys do too. And that's great. I think I should change this, make it a little bit more visible. Appearance. So there, I accidentally chose 13, and I was superstitious, so I changed it to 14, which is pretty not very liberal of me. Yeah, so this is our studio. It looked like this when we started. I clicked this little icon here to get a panel here where I can write R commands. And what I wanted to do was, Barry, I'm getting to your question, believe it or not. I wanted to go to this document, and it says week2 demo.r. It also says it at the top of the document in the key resources. It takes us to this little gist on GitHub. I'm just going to like select it all with my browser, Control C, and go back to RStudio and paste it in there. And I'll just follow along with this a little bit. So I guess I just wanted to introduce, you know, in many ways, I can see I put way too much stuff into this this pop-up workshop, but so we kind of got oriented, right, with the workspace. We had the different workspaces. We had this data tab. We ran the script. It wrote data from a Google bucket, and it wrote data to a Google bucket. And there were all of these like graphical elements that we could use to manage things. And I wanted to introduce, so this is like part 2 of my presentation. I wanted to introduce this AnVIL package for scripting your interaction with that graphical environment in a way that probably empowers you as a user. And we're using it inside RStudio, but we could just as easily be using it in these other notebooks. So in this script that I've copied in, I'm just going to maybe just walk through it very quickly. The first couple of lines say, if you haven't already, in your runtime, if you haven't already got the AnVIL package installed, then install it. And so it does that, and I hope that you guys follow along. It pulls down several dependencies. You can see, just as last week, it's using the binary version of these packages provided by RStudio. So when it's retrieved the packages, it doesn't actually have to compile them, it just installs them. And that's true for all of the packages except for AnVIL. And AnVIL it installs in the same old way. And then it says, do you want to update these old packages? And I'm going to say no. And that's it. I now have the AnVIL package installed in just a few seconds. So I've just run this command here. And now what I wanted to do was introduce maybe some of the key functions in the AnVIL package. And again, the vignette of the AnVIL package could be a good source of information. There's this command called AV-Workspace. And Barry, we're almost at where you're asking about. And what it does is it figures out the workspace that this runtime was run in. And so it says, okay, now I know the workspace that I'm in, that I'm actually running in. But I've also added 2 more workspaces. You can see the workspace consists of 2 components. This is called the namespace. And you can see it's like the billing project. The namespace, and then this is the workspace name. So the namespace and then name. So this is the I think we'll probably only get to this in any kind of detail, but the HCA is just the workspace that we were just visiting through the graphical user interface. And then the 1,000 genomes is another workspace that we could navigate to, workspaces, look for this 1,000 genomes high coverage workspace. So like, we're just creating references to these workspaces. So right now I'm in the workspace.
I'm sorry, can you explain how we find the names of the workspaces, like how we would determine what they are?
Yeah, so I'm going to go to, hopefully that I'll be able to manage here. So like under hamburger workspaces, I'll go and look at the 1,000 genomes. And I'll look under public workspaces, and this is the workspace that I was interested in. I'll just click here on the 1,000 genomes, and I'll actually just look at data, and again, pay no attention to these warnings and errors, and look at one of these tables here. So like this is a different workspace, and actually up here at the top it says workspace, AnVIL data storage, 1,000 genomes, blah blah blah. So this middle part after the workspace, that's what we're naming it after. AnVIL data storage, 1,000 genomes, high coverage, 2019, and it actually shows up here in the URL as well. So that was my way that I'd find that. And actually, when we were going into this workspace, just before we clicked on it, all of that information was available as well. Sound good?
Thank you.
Yeah. Okay, great. So like right now we're in the my pop-up workshop workspace, but now we're in the HCA Optimus pipeline workspace. And in the HCA Optimus pipeline workspace, we saw those tables of data, and we can discover what they are from this workspace. There were 3 tables: a participant table, a sample table, and a sample set table. And then we can actually go and retrieve those. So instead of, we were in workspaces, HCA, public workspace, pipeline, data, sample. Instead of, looking at them in this data browser, we could instead say, hey, I know about this workspace, and I've discovered that there's a table called sample, and so why don't I just retrieve it into R? And then now that I've got it in R, I can do with it whatever I like. I've got the FASTQ files. I've got whatever information it is in the sample table, and so I can do with it what I like. So, for instance, there's a column called participants, so I could find out that there are 6 participants with this value, 2 with this, and 2 with that. So actually, we can interact with the components of the workspace through the AnVIL package, which is pretty neat. I'm just going to go to the 1,000 genomes workspace and switch over to the workspace over here, find the 1,000 genomes. 1,000 genomes. If we go for 100, we'll probably find 1,000. Look at the data. And then there are these tables, like there's this pedigree table, and there are 32, in the 1,000 genomes workspace, there are 3,200 genomes, of course. And we can see what population each of the individuals are from and their sex. And maybe we're interested in how many individuals of these different sexes there were from each of the populations, just kind of standard data manipulation things. So we go over to the 1,000 genomes workspace, look at the tables that are there, participant, pedigree, sample, and then let's look at the pedigree table here. Here's the pedigree table. So it retrieves the 3,200 rows from the pedigree table. We've got the population, the sex, and then we can do whatever manipulations we want. And I'm gonna use this. This is tidyverse kind of syntax. It might be kind of new to people who aren't who it might be new to you if you're not into the tidyverse, but it's kind of a different way of writing R functions. You have this function, and then this is an operation that says take the stuff on the left-hand side and use it as an argument to the stuff on the right-hand side. So this says take the result of this, which is this data frame. It's called a tibble in tidyverse. This data frame, and feed it into this function count, which summarizes the number of individuals with a particular value of population and sex. And then this command says, and then make it into a table with two columns, one for sex and one for each sex. And so it says, in this population, there were 57 individuals of sex 1 and 59 individuals of sex 2. And in this population, there were 33 and 41 and 60 and 71. So just illustrating that we can actually interact with the data in a very friendly and familiar way. Also, I have no idea which is sex 1 and sex 2. Any votes? Is that a convention in the 1,000 Genomes project? Sex 1 is always, maybe it's not important, but it must be important. Yeah, so I don't know. Anyway, so for instance, you could recode sex from 1 and 2 to male and female once you figured out what the right code was, and then you could add it back to the table and then have something sensible as the labels on the table. All right, back to the HCA workspace, and so that's kind of fun, right? Like I was visiting the 1,000 Genomes workspace, and now I'm back in the HCA Optimus pipeline workspace, and I pulled down the sample set table, and here it is. And one of the outputs of the workflow was a Loom output file, and the workflow said, hey, update this sample set table with the name of the output file that resulted from this workflow. So when Elizabeth ran this workflow back in January, it took those FASTQ files, ran it through the workflow for each of these sample sets, and then produced a Loom output file. And here's the Loom output file. We can probably make it look just a little bit better here. So let's see, what have we got? We've got our tibble. We can at least select just the Loom output file column. So you can see GS, that means that it's a Google bucket. And actually, this part of the Google bucket, you'll see that it's common across each of the output files. It actually refers to the workspace. So let's go back here. Go back to the HCA. I feel like I've totally, when a 6-year-old starts getting excited about their trons or whatever, their artificial world they're living in, they sort of start talking gibberish about the characters in their world. But so I feel like I'm at that stage of the presentation now where I'm pretty much talking gibberish. Andrew shook his head, so that's good. That's great. So, okay, so the workflow produced, actually added this file called Loom output file, and it stored it in a Google bucket. And you can see the Google bucket starts with SD-28 blah, blah, blah. And then the next element of it is B63 blah, blah, blah. But actually that Google bucket, part of the workspace is a Google bucket. So this particular workspace has its own Google bucket. The 1,000 Genomes workspace has its own Google bucket. The workspace that you cloned has its own Google bucket. And so I'm not sure that this is going to work. This seems to be one of those things that is just being a little bit problematic at the moment. But actually, and you can see in this notebook, in the notes here, actually what happens is each time there's a run of the workflow, it creates a whole bunch of entries in the Google bucket, one of these for each of the workflow runs. And somewhere inside this, you can think of it sort of like a file system, but it's an object store. But somewhere inside each of these is the Loom file that we're interested in. And so what this is saying is that if we drilled into that bucket and looked at this location, and then actually, I think we have to go even further here, and actually pulled out these guys here, drilled down, we'd eventually find the Loom file that was the output of the workflow. So the workflow ran, processed the FASTQ files, produced this Loom file as an output, and then stored the Loom file in the Google bucket associated with the workspace. So we kind of got the whole analysis. We know where the inputs are. They come from that data tab. We know what was run, the script that was run, and we now have the outputs from the workflow. So what I wanted to do was, what did I want to do? Oh, I wanted to move the fact that these Loom files are in a Google bucket is great at some level, except that none of our standard software knows how to read from a Google bucket. We know how to read from a local file system, but we don't know how to read from a Google bucket. So what I wanted to do is I wanted to move those files from the Google bucket to my local file system so I could operate on them. So that's what I do. I say, I'm going to create a directory in Rstudio/Loom. So this is on my persistent disk. And you can see actually over here, I've got, here's the home. I just created that directory. And then I'm going to take this vector of 3 Loom files, and I'm going to use this function called gsutil for the Google Cloud Storage utility, TP, and I'm going to copy from these 3 files from their location on the Google bucket into my local hard drive so that I can process them. And this gsutil CP is from the AnVIL package. So I'll just do that. Oops. I'll do that. And we'll see that it takes a second or so. But actually, it copied 500 megabytes of data from the Google bucket to the local drive. And all of that was in the cloud. And it doesn't cost us anything to move data in the cloud like that. And it was also pretty fast. So, and then when we look in our directory that we created, we can see we've got these 3 files, the 3 Loom files, ready to be processed in our R session. So we've kind of, this is the point in the workshop where really, looking up at the time, it's 12:58. I've done about 2/3 of the material I was wanting to cover, and I really just have to say, okay, that's really great. I hope you found that really, really great. So I'll just say, that's totally cool. We can do other things with our Google bucket, like we can back up the files that are on our persistent disk into the Google bucket associated with our workspace. And it's called backup, and that might be sort of like think of it as like a security thing or something like that. But actually it means that whoever has visibility in the Google bucket, so in the workspace, would now have access to the scripts that I'd written. So it'd be a way of sharing my scripts with my lab mates, for instance. I could write them to the Google bucket, and I could likewise restore them from the Google bucket to my local file system. So you can think of this as a way of kind of locally publishing your work to the Google bucket so that people can share it across the workspace.
So Martin, with that line on line 56, we don't have the directory, your home area scripts.
Yeah, right. So what I did actually was, I guess if I were taking a more leisurely walk through this, I would have actually, so this script that I copied from the GitHub gist, I'd actually save it as create a new folder, scripts, save it as week 2 script -r.r, and then I've got this script directory here, and then it would exist, and I could store it to the Google bucket.
Perfect. Thank you.
It looks like I've done something not quite right. But.
Martin, could I ask what happened with that AV Workspace HCA command? It seems like you can just pop around between different workspaces and copy files from them into your runtime, into your persistent disk.
Yeah, cool, eh?
Is that a feature that's going to stay, or is that something that is going to go away with this notebooks being tied to runtimes, or
it'll
tied
stay
to workspaces?
Yeah. So actually, you said you popped around, but you didn't actually. You only popped around to the workspaces that you had access to. So you can't pop around to a workspace that you don't have access to. That's hard to show, but you can't do that. So it's not like you're doing anything that you don't have permissions to do. So I think it's good. It's also incredibly powerful, right? Like now we have the 1,000 genome samples, all of that information available to us just within this workspace, even if we're working on something quite different.
And are there any costs incurred when that copying occurs, or is there only costs for us for the persistent disk?
Yeah, so this is the way that cloud providers work. If you copy within the cloud, it costs almost nothing, minimal. The real cost is when you do an egress, so when you move from the cloud. So like if you took those 1,000 genomes files, the FASTQ files, and you downloaded them to your local laptop, that would be expensive, measurably expensive. Whereas if you copied them to the persistent disk, you wouldn't. There'd be very minimal egress charges. There wouldn't be egress charges. They'd be like data transfer charges within the cloud, so they'd be inexpensive.
And it was so fast, it seems like, to the extent that you might want to just delete that disk if you're going to put it aside for a couple of weeks.
Yeah, maybe that's a good strategy, sort of money management. I mean, the disk costs, it's a small disk in some ways, like 50 gigs is a pretty small disk, so we'd fill it up pretty quickly. But it's not super expensive either. Actually, I just ran into this little error here. I tried to back up my scripts folder, and it says, hey, I was trying to do this command, and it says, actually, this user, and you can recognize some part of it as the deep bioconductor, does not have storage access to wherever it was trying to store it to, and is actually trying to store it to the HCA, the bucket under the HCA Optimus pipeline, which I don't have access to. And so what I did, quietly, was I switched back to this workspace, and then when I try and do the backup, it'll be successful. And copying my script up to, I should be able to see under the files, I should be able to see my script here. And anyone who was able to see this workspace would be able to see the script. All right,
excuse me.
we're kind of at the end of time, so I got to just stop sharing, but I continue to talk here. So, yes.
Martin, it'll bug me if I don't ask it, but
yeah.
So to navigate between the different cloud environments that you might have started or not have started, and to list them, say I've launched Jupyter 1 and an RStudio 1, is there a way on the web interface to see them or go through them? I know they're associated with workspaces, but
yes. So of course, I'll just start sharing the screen now that I stopped sharing. So like under the hamburger, and there's this entry called Cloud Environments, and I think that Terra is having problems with accessing this information, but it'll show if you've got an RStudio. So these are the runtimes that I've got going for me now, and I can delete them and find out the details about them as well.
Fantastic, thank you. And then those persistent disks, are they the same as those Google buckets you were mentioning that are listed underneath our workspaces? Or are they a distinct?
They're distinct. Like the Google buckets, so it's like they're an object store, and whereas the persistent disk is like a standard, disk that you would have had in your laptop. And so like on the persistent disk, like in our RStudio session, now I have to find my RStudio. Like over here in this files, this is the persistent disk, and I can do everything that I do on a file system. I can do here. I can navigate around, and these files I can read in using the appropriate commands and so on. So this is the persistent disk, and I can do all those sort of file-like things. But the buckets, which are under these files, they're just like object stores, and basically all you can do is copy stuff from here to somewhere else, and the reverse. So you can't.
So the general workflow is the important stuff you write to these buckets and keep them there. You don't keep them on the small persistent disk, or the name persistent is confusing me. Is it persistent like it's always there, or it's not there and it goes away when I?
Yeah.
What's the thinking?
Yeah. In many ways, I think you should, like persistent disk, we should rename it, and it should be like like a local disk or something like that, like a computable disk or something like that. It's the thing that you can perform computations on, but it's relatively expensive. Like if you were storing like your own FASTQ files, you'd much rather store them in a Google bucket than on a persistent disk, because the persistent disk would be tens of or even hundreds of dollars a month for like a large chunk of data, whereas the object store would be very cost effective. So like the Google bucket would be very cost effective. There's also this other element about visibility. So like in your persistent disk is really only visible to you. It's tightly coupled to your RStudio instance or your Jupyter instance. And the way that you'd share, say, data with lab mates would be you'd create a workspace and you'd put the shared data into the Google bucket on the workspace. Like a group of you are analyzing a particular experiment. So you put all of the FASTQ files in the Google bucket, not just because it's cost effective, but that way everyone has access to the same files.
And then the bucket storage, like if we write some files here, we're getting a charge for that storage, like same as you would with an S3 or whatever.
Yes. So we'll talk. That's a great question. So a little bit later we'll talk about billing. I can't remember it, probably towards the end, but basically everything you do, you're getting billed for, and so the storage on the Google bucket costs you money based on the amount of data that's stored there. You'd only start to worry about that when you're like it would only be a concern when you started to store terabytes of data. That would sort of be when you'd start wanting to watch the cache register.
Just to be a pain, I tried copying one of the 1,000 genomes VCFs, and I got one note and one exception. A note that I should learn about CRC mod in GSUtil, and an exception that it requires integrity checking with CRC32C. But my CRC mod installation isn't using. Right.
So actually, the first one I've seen, the note I've seen before, and it points to like the it's pretty interesting, right? Like the runtime, this particular runtime is based on a Docker image, and the Docker image includes this piece of software called the Google SDK Software Development Kit. And the Docker image actually needs to be updated to avoid those errors. So that's like a bug fix that needs to be introduced. And probably there'll be a new release of this runtime probably in early June, and it'll probably be updated then. Yeah.
Nice. Thank you, Martin. That was awesome.
Yep. Thank you, Martin. Very, very good. Thank you.
Yeah. Great. Thanks so much, and I'll see you all next week.
Until next week, all. Thanks very much, guys.
Thanks, Martin.
See ya.
See you.